Assalamu alaikum, dear students. In this muscle physiology lecture, we will talk about the skeletal muscle contraction, and specifically, we will talk about the thick and thin filaments. We will discuss about in detail that what is actin, what is myosin, and what are the other filaments which are present in the sarcomere, what are the parts of sarcomere, and how contraction, relaxation occur. So, sarcomere, which is actually the structural and functional unit of skeletal muscle, is made of the actin and myosin filament. Actin, which is the thin filament, and myosin, which is known as the thick filament. So, this is the sarcomere, which is actually, as we discussed, the structural and functional unit of the skeletal muscle. If we look at the sarcomere, so we can see there are some thick filaments. These thick filaments, they are known as the myosin, and between these thick filaments, he has a narrow band, and these thin filaments, they are known as the actin. So yeah, so these are the actin, these are the myosin, and these are the cross bridges which through which there is contraction or relaxation of the skeletal muscle. So we will discuss each of them in detail about the myosin, about the actin, and as well as about the other components of the actin filament. So each muscle fiber that is made of several hundred to several thousand of myofibrils, and the thick filaments in these myofibrils, they are known as the myosin filaments, and the thin filaments, they are known as the actin. What are the cross bridges? Cross bridges, they are small projection from the myosin filaments. These interact with actin filaments for the contraction. So remember that where are these cross bridges? They are present on the myosin filaments on the peripheral side of the myosin filament. There is no myosin, there is no cross bridges, and the center of the myosin filament. So this is actually a molecule of the myosin filament. You can see here a single myosin molecule has a tail and a head. The head has each head has two sides. One is the actin binding site, and the other is the ATPase site. There is the side where the enzyme ATPase attached, and this is actually the binding site where it contract with the actin, and hence it contract or relax the muscle by just contacting with the actin with this actin binding site. Now, what is Z disc and sarcomere? Z disc is the point of attachment of the actin filaments. We can see here these are the thin filaments which are present between the cross bridges, and this is actually the Z disc, and this is also the Z disc. So the thin filaments, the actin, they are attached to the point which is known as the Z disc. Extend in both direction, and the both sides, the thin filaments, the actin, they extend on the both side of the Z disc. Filamentous protein different from actin and myosin. So the Z disc is also made up protein. So it is made a protein in nature, and that give actually the striated appearance to the skeletal muscle. Now, what is the sarcomere? Portion of the myofibril or the whole muscle between two consecutive Z disc is called as the sarcomere. For example, this is one Z disc and this is the next Z disc. So each and everything inside these two Z discs, it is known as the sarcomere. If there is, this is the one Z disc and there is another Z disc, so between these two consecutive there would be another sarcomere. So it means each the muscle, the skeletal muscle is made of sarcomeres, and hence there is continuous work of each sarcomere in the contraction or relaxation of the muscle. The length of each sarcomere is almost about approximately 2 micrometer. Actin filaments completely overlap the myosin filaments, as we can see here. And during the contraction, there is no change in the position of the myosin. Myosin in the, this is actually the relaxed position of the muscle. This is the when the muscle contracted. So if we look at the space occupied by the myosin, this is similar in both the contracted muscle and the relaxed muscle. There is eccentric contraction and concentric contraction. So what are the changes which occur during the contraction of muscle in the sarcomere? So there is shortening of the edge zone. What is edge zone? Edge zone is the distance between two actin filament. We can see here, and चूंकि यहाँ पे एक्टिन नहीं होता, तो इसलिए यहाँ पे क्रॉस ब्रिजेस भी नहीं होते. अब क्रॉस ब्रिजेस किस लिए होते हैं? ताकि ये इस एक्टिन के साथ अटैच्ड हो जाएं और इस या तो इस एक्टिन को एक दूसरे के करीब लाएं या एक दूसरे से दूर लेके जाएं. तो ये इनका काम होता है इस क्रॉस ब्रिज का. अब चूंकि इस ईरा में कोई एक्टिन फिलामेंट नहीं है, तो इसलिए यहाँ पे मायोसिन पे कोई क्रॉस ब्रिज नहीं है. सो नाउ इट इज वेरी क्लियर टू ऑल ऑफ यू that what is the reason that in the center of the myosin there is no cross bridges. So the answer is that the cross bridges, they are further attachment with the actin, as there is no actin in the center, so no cross bridges. So पहला जो चेंज इसमें आती है ड्यूरिंग द कंट्रैक्शन ऑफ मसल इन द सर्कुलर, देयर इज शॉर्टनिंग ऑफ द जोन. अच्छा, एक और बात यहाँ पे, सर्कुलर इज मेड ऑफ द आई बैंड एंड द ए बैंड. सो व्हाट इज द ए बैंड? A band is that area of the sarcomere in which the myosin is present. So from one end to the other end, total length जो myosin की होती है, that is known as the A band. As we can see here, this is the length of the myosin. So यहाँ से लेकर यहाँ तक, this is the A band. So हम ये कह सकते हैं कि मसल अगर कंट्रैक्ट, उसकी इसेंट्रिक कंट्रैक्शन होती है यानी रिलैक्स होता है, लेंथन होता है, या कंसेंट्रिक कंट्रैक्शन होती है, मसल शॉर्ट होता है, A band जो है ये remain the same. There is no change. You can see here there is no movement by the myosin. On the other hand, there is another I band. I band mean this is the end of one myosin and the start of the next myosin of the next consecutive, the next sarcomere. This distance is known as the I band. So we can see here, uh, this is the end of the A band. So यहाँ से लेकर the end of the start of another A band, this distance is known as the I band. हम इसको इस तरह भी कह सकते हैं कि I band is actually that part of the sarcomere, two consecutive sarcomere, in which there is no myosin filament, only the actin filament is present. अब इस जोन क्या है? इस जोन एक्चुअली ए बैंड का ही हिस्सा है, लेकिन इस जोन का वो हिस्सा जिसमें सिर्फ और सिर्फ मायोसिन होते, एक्टिन नहीं होती, दिस इज नोन एज द इज जोन. और इस जोन को अगर हम निकालें तो रिमेनिंग ए बैंड में एक्टिन भी होती है और मायोसिन भी होती है. यानी सपोज यहाँ से यहाँ तक दिस इज द एज जोन. इस एरिया को जो इज एक्चुअली द एरिया ऑफ द ए बैंड, बिकॉज द मायोसिन इज देयर, इसमें एक्टिन भी है, मायोसिन भी है. So this both area, the edge zone and the era where both are present, that is known as the A band and I band. So there is shortening of the I band as well as there is shortening of the edge zone, but there is no change in the A band. So remember this point, during the contraction when the muscle is going to shorten. So this shortening is because the shortening of the edge zone and the shortening of the I band. Then the midpoint of the edge zone, this is known as the M line. And this dark blue purple color, this is actually known as the Z disc. नाउ क्वेश्चन ये राइज होता है कि इस सार्कोमेयर में एक्टिन और मायोसिन को अपनी जगह पर ये क्यों इतने स्टेबल होते हैं, ये वहां से इनकी इंस्टेबिलिटी क्यों नहीं होती? सो द आंसर इज द टाइटिन मॉलिक्यूल. So basically, titin molecule a contractile molecule नहीं है, just like the actin and myosin. It is another protein in nature which actually provide strength and just provide anchoring to the actin and myosin, specifically the actin molecule. So they are also filamentous in nature, and they are springy and one of the largest protein molecule in the body. Now, each and everything that we have discussed in the that is known as the sarcomere, but the fluid present inside the sarcomere is known as the sarcoplasm. So we can say that the cytoplasm of the sarcomere there is known as the sarcoplasm. So that sarcoplasm they contain potassium, magnesium, phosphate, large number of mitochondria. Parallel to myofibril, there is ATP. This is the myosin molecule structure which we have discussed as well in the previous slide. It's made up two heavy chain and four light chain. We can see here one, two, three, and four. So four light chain and two heavy chain. The two heavy chain like a helix, they just wrap around each other. This is known as the head. This is the tail. So this is the structure of the myosin molecule. Each myosin molecule filament that is made up 200 myosin molecule. Remember this point that how many myosin molecule are present in each myosin filament? 200. Each myosin molecule is made up the six polypeptide chain, in which four are the light and two are the heavy. We can see here these two are two and these two that make four light chain, and this red and blue these are the two heavy chains, so they make the six polypeptide chain. Heavy chain wrap spirally around each other from the double helix, which makes the tail of the myosin molecule, and two pairs at one end, which having two side. One is for the ATPase, the other is for the actin. Two light chain to each heavy chain. This is the myosin filament then with attachment with the actin filament. These are the cross bridges, and through the cross bridges they can either um, make the actin to come close to each other or just move them away from each other through the movement of this cross bridge. This is the body of the myosin filament, and these are the cross bridges of the myosin filament. These points are known as the hinges, while the entire limb is just known as the arm of the cross bridge. So tail of the myosin molecule bundled together to make the body, while the head hang outward away from the body, which part of the tail also hang out, which is known as the arm. This is actually the arm, and this is that head. Arm plus head that make the cross bridge. So actually, cross bridge is part of the myosin, and it has the binding site for both the ATPase and the above and below present actin filament. Each cross bridge is flexible at 2 points hinges. One is when it is attached with the myosin body. हम देख सकते हैं कि यहाँ पे दो hinges पॉइंट की गई हैं. तो एक फ्लेक्सिबल यहाँ पर है when the cross bridge arm जो है ये बॉडी के साथ अटैच होती हैं. दूसरा जो इसका फ्लेक्सिबल या है that is actually the hinge that is between the arm and the head. This is the head and this is the arm, and arm is actually the part of the tail. As we have discussed here, the part of the tail also hang out, and that is the arm. So arm plus tail, that is equal to the cross bridge. Length of the myosin filament is almost 1.6 micrometer. No cross bridges in the center, and that is actually zero point 0.2 micrometer, and that is actually known as the edge zone. Each filament twisted cross bridges extend in all direction, angle of 120 degree. Remember this again, how much degree is required and how much degree the cross bridge can move. So this cross bridge, if they can move from here to there, they can move at the level up at the angle of 120 degrees. So they have the two binding side, the myosin at the base side and the actin binding side. These two are the heads. This is the tail. These are the myosin molecules, the bunch of myosin molecules. Myosin head act as ATPase enzyme and essential for the contraction. This is now the next actin filament, which is again made of the troponin complex. The tropomyosin are the F-actin. Three protein components are present in the actin filament. One is the actin itself, the tropomyosin, and the troponin. This is then the actin strand. This is the G-actin. And this is actually the black spot on each G-actin, which means the globular actin. This is the myosin binding site. As we have observed, there is a binding site on the head of that myosin, which is known as the actin binding site. Similarly, there is also a binding site on the actin known as the myosin binding site. So this is the myosin binding site and myosin पे actin binding site है और actin पे myosin binding site यानी myosin जो है actin के साथ अटैच होगा और actin myosin के साथ अटैच होगा. So ये जब G actin सारे के सारे ये combine हो जाते हैं ये the actin molecules so they make the double helical actin strand. तो यहाँ पे फिर है हम देख सकते हैं कि यहाँ पे ट्रोपोमाइसिन भी प्रेजेंट है और ट्रोपोमाइसिन के साथ साथ ट्रोपोनिन कॉम्प्लेक्स भी प्रेजेंट है. So these are the actin molecules. This is the binding site for attachment with myosin cross bridge. And जब ये बहुत सारी G actin बनते हैं so they make a double helical structure known as the actin helix or the actin filament. This is the G actin, this is the F actin. उसके बाद फिर ट्रोपोमाइसिन एज वेल एज ट्रोपोनिन ये दोनों भी कंबाइन होते हैं सो दे मेक द थिन फिलामेंट कंबाइनली वी कैन सी हियर देयर आर this is the actin helix, this is the tropomyosin and this is the yellow one is the troponin. So actin helix plus tropomyosin plus troponin that equal to the actin filament. Now the actin we have discussed that F15 and G18. So F15 is basically the filamentous actin molecule and G is the globular actin. Backbone of the thin filament. Backbone mean if there is no F18, there can be no actin filament. So the actin filament, the main component of the actin filament is the F18 molecule. And that is present in two strands which are round in a helix. G18 molecules, each strand of F18 helix is composed of polymerized G18 molecules. So the F18 molecule is a polymer of the G18 molecule as we have discussed, and attached to each G18 molecule is one of molecule of ADP. Length of the each actin filament is equal to 1 micrometer. Now the active site of actin filament, there are ADP are the active site on the actin filament and head of the myosin filament bind with these actin site. Active site on the two F15 strands of the double helix, one active site. The actin filament is also consists of the tropomyosin as well as another protein, and the length of the tropomyosin is almost 40 nanometer. and it is wrapped spirally around the side of the F-actin molecules, lie on the top of active sites in the resting state. और जब यहाँ पे रेस्टिंग स्टेट से निकला कि यहाँ पे कंट्रेक्शन होती है तो फिर ये वो बाइंडिंग साइट जो है ये उसको छोड़ लेता है. So troponin complex attached intermittently along the tropomyosin molecules, three subunits, troponin 1 affinity per actin, troponin T that is affinity for the tropomyosin, and the troponin C which is affinity for the calcium. So the troponin is again divided into three subunits, troponin I, troponin T and troponin C. So this is actually the troponin complex. This is the troponin complex. One is the troponin I, to the other is the troponin T, the other is the troponin C. This is the tropomyosin, and the green one, this is the F15, and now it is the actin. Binding of myosin head with the active site in the presence of calcium, because calcium is very necessary for this contraction, and hence their attachment of the that myosin head with the active binding site here on the F18. That was all about the thick and thin elements of the sarcomere. We discussed them. What is sarcomere? What is Z disk? What is the H zone? What is the A band? What is the I band? The actin molecule, the myosin molecule. What is the main function of titin? So that was all about today's lecture. Thank you so much to all of you.